Okay.
So let's finally begin. So hello everybody. I am very grateful for the opportunity to give a talk on our community. And really, my dream is that once we meet offline and could communicate not by using uh, difficult apps and time zones. So, in my presentation, I would like to make a small review of our research project that we do with my team in our institution. So I represent my institution, the Kovalevsky Institute of Biology of the Southern Seas. It's one of the eldest research institute of Russian Academy of Sciences, and it now we are 153 years old, and our institution has started as one of the first biological stations in Europe, and it was the first biological station in Russia. And today we occupy the same building, and we have 15 departments and laboratories in our institution, and our institute, our scientists make research in the field of biology, ecology, physiology of marine organisms, radiobiology, and the development of biological active compounds from marine organisms. And I am the member, I am the head of laboratory of ecological immunology of aquatic organisms, and we are one of the youngest departments of our institution. We have only 10 members in our team, and only 5 of us are PhD. The other 3 are PhD students, and 2 of us are master students. So we are really a young team, and we are only at the beginning of our scientific way. So please don't judge us too strictly, too seriously, because our lab works only less than 3 years. But the aim of our lab is to study all principles and to make a fundamental research in the topic of health of commercially important bivalve species, with the focusing of the prevention of diseases on shellfish farms, since diseases are considered as a major problem for global aquaculture in the 21st century. And we study the effects of various environmental factors on the immunity of bivalves that may occur on a shellfish farm. And also we study how the aspects of physiological stress in bivalves and their general term of bivalve health. And of course, we study the oxidative stress and how the oxidative stress develops at cellular level in bivalve mollusks upon environmental stress, and which are the range of the adaptive potential of bivalve mollusks or during the various environmental stressors. In the recent years, we are focused on the impact of global climate change on the bivalve physiology. We all know that there are changes in our climate on the planet, and there are several problems that occur in aquatic environments. For example, here I mentioned only a few factors, but they are one of the majors. For example, changes in temperature induce a decrease of oxygen solubility in water and also enhances metabolic processes in ectothermic organisms, which induces environmental hypoxia in aquatic systems and functional hypoxia in organisms. Also, change in sea levels that increases or decreases in different regions cause periodic short time or long time fluctuations of ocean salinity, and osmotic stress is one of the major stressors for aquatic organisms that raise the questions of their survival in altered salinity, in regions with altered salinity. And also higher emission of carbon dioxide cause ocean acidification, and this is one of the top research research topics, especially in Bibalves, since they are considered like sensitive organisms due to the problem of with their shell that become fragile in acidic water. So take into account these problems, we started like several projects in our lab, in our team, that was funded by the Russian Ministry of Sciences and Education, Foundation of President Grants, and Russian Science Foundation, where we decided to compare the effects of salinity, acidification, and hypoxia on commercially important bivalve species. Since bivalve aquaculture occurs in coastal aquatic systems that possess more fluctuating conditions compared to open water, and moreover, bivalve mollusks are farmed in conditions of open aquatic system, and there are no in Russia there are no special feeding, there are no special usage of so they are in open water, so we can only rely on the stability of the conditions where these mollusks are farmed. And we decided to compare how these environmental factors affect immunity and the oxidative stress in two major commercial species for Russia and globally cultivated bivalves is the Pacific oyster and Mediterranean mussel. These species are different by their tolerance to hypoxia, but they possess close range of salinity in their natural habitats. And we don't know anything about their tolerance to water acidification. And here, in my further slides, I will show you the results of the experiments that we conducted to find the answers on the questions, on these questions. So here in our group, we don't use natural experiments, and it's our closest future, I hope. And therefore, all the results that will be present here were obtained through the experimental modeling of hypoxia, salinity fluctuation, and acidification. And so I will show you, it's like three large experiments that we did in recent 2 years, and some results are still under analysis. For example, here I show you that in the Pacific oyster we can see the whole picture. Oh, maybe I will use here. Yes, it will be easier. So first of all, we decided to evaluate how our species acclimate to salinity fluctuations. Since the Pacific oyster is the introduced species in the Black Sea region, and the salinity of its farming in the Black Sea is lower than its natural salinity, since the salinity of the Black Sea is 18 PCU. And the Mediterranean mussels naturally occur in the Black Sea. It's like a massive benthic species, and so natural salinity of Black Sea is comfortable for this species. So we made a salinity experiment where we placed the mussels and oysters into the water and started to change the salinity in the aquarius with the speed 2 PCU per day. And when the salinity levels reached 6, 10, 14, 24, 30, and 40 PCU, we maintained mussels and oysters at these conditions for 2 days, and then sampled the parameters. To study, to evaluate the changes of the oxidative stress at cellular level, we sampled hemocytes where we measured the levels of reactive oxygen species, and we also measured the degree of DNA damage. And we also chose the gills as the most sensitive target tissue to evaluate the level of antioxidant activity of antioxidant enzymes, and also we evaluated the levels of oxidation of macromolecules by T-bars level and protein oxidation levels. So what we obtained in this experiment here, the general scheme is presented. It was similar in mussels and oysters. And what about the mortality? We found that during the acclimation to unfavorable salinity level, we found that oysters in groups, the mortality level reached 16 to 80%, and it was so high that we couldn't even sample tissues and cells for the analysis. There was not enough statistics. And so we concluded that oysters are extremely sensitive to salinity fluctuations in this salinity range. And for mussels, the picture was another. We found that mussels acclimated to salinity, to unfavorable salinity conditions pretty well, and the mortality was low except one group where salinity level reached 40 PCU, and here the mortality was very high. So we once again we couldn't sample and to make any analysis of their functional state. But the mortality, I think, tells us everything, that it was out of the range of acclimation possibilities. But in the groups that were acclimated to salinity from 6 to 30 PCU, we have obtained several interesting results. For example, we found that salinity changes promoted rose formation in hemocytes, and as you can see from this graph, the extreme salinity levels were associated with the highest levels of rose production by hemocytes. And similar picture was obtained when we analyzed the level of DNA damage in hemocytes. You see that distant salinity levels induced very intense damage of the DNA in hemocytes of mussels. And when we evaluated the antioxidant capacity in gills, we found that that in gills the picture was not so bad, since the activity of the major antioxidant enzymes decreased only at salinity 24 PCU, so it was hypersalinic conditions for mussels. And also we found at this salinity level we observed the decrease in 3 measured enzymatic activity. So all three enzymes were depleted at these salinity levels. And also we observed that the lowest hyposalinic stress in mussels was also associated with a decrease of the activity of superoxide dismutase. However, these fluctuations of activity of the antioxidant enzymes were not associated with activation of lipid peroxidation in gills, but we found that the lowest salinity level 6 PCU, was associated with an increased protein oxidation levels. And we found it in all 3 groups of measured proteins. So, what is the conclusions on this experiment? We found that mussels pretty well acclimate to salinity levels about from 18 to 14 salinity. So they and till 24 salinity, where they we didn't found not so much oxidative stress markers. However, and in all groups, we found that the hemocytes are damaged by by oxidation processes. And this here we can conclude that hemocytes are really more sensitive to oxidative stress compared to gels, and their antioxidant capacity is really low. And this result is very important since hemocytes are major immune effector cells for bivalve mollusks. And here, since I show you only the results on the oxidative stress, but we also find that the immunity responses were also depleted. So salinity fluctuations cause immunosuppressive effect on bivalve mollusks, and also induce oxidative stress in hemocytes. But in gels, the capacity of the antioxidant system is really high, and I think we think that the in this case, in the condition of osmotic stress, gels are really but a high adaptive capacity to osmotic stress. But the interesting result is that hemocytes are really vulnerable because you know that bivalve mollusks do not acclimate like at system organismic level to oxidative stress. For example, how it's how it works in fish. And their internal osmolarity corresponds to external osmolarity, and therefore the osmotic stress occurs first of all at cellular level, and we see that the capacity of cells to neutralize oxidative stress during the osmotic disturbances are really low. Especially when we say it about hemocytes. And for the general conclusion for the Pacific oyster is that for its farming in the Black Sea, Pacific oysters are really vulnerable for salinity fluctuations. Maybe this is because they are transferred from the Mediterranean Sea, and we didn't have oysters in our farms that were born in the Black Sea. All spot is bought from the other regions. Oh, so the next question in our lab was how water acidification at different oxygen regimes affects the oxidative stress markers in commercial bivalve species. And in this part of research, we just finished this project this winter, and so not all results are obtained, and I will show you only the data on the Mediterranean mussel. So we also again we did experimental modeling of water acidification. We decreased water pH till 7.3 level, and maintained mussels for 8 days at these conditions, and sampled periodically the parameters to see the dynamics of changes in the organism. And we also verified how the combined effect of acidification and hypoxia maybe there is an interaction between these factors because in general in nature acidification usually occurs with the combination with hypoxia, and acidification combined with normoxia is like a kind of a toxic effect when we, for example, some acid compound enters aquatic environments. It's like an anthropogenic effect, and this is the case of global climate change. And similarly, we evaluated the levels of reactive oxygen species, the level of DNA damage in hemocytes, and similar parameter in gills. And what did we see? We see that the antioxidant response in gills was really low. So we didn't find any changes in the activity of superoxide dismutase and catalase, and we also didn't see any markers of the oxidation, tissue oxidation. However, we found that the activity of catalase decreased compared to the first day of exposure. So we see that the period of exposure to low to acidified water affects the activity of catalase. And so we may suppose that long time exposure to acidified water may cause a decrease of the activity of the antioxidant enzymes. But during the period of 8 days, that in general corresponds to periodic acidification in the Black Sea regions, everything was pretty well. And similarly, we didn't observe any damage in DNA in hemocytes, and the level of intracellular ROS production even decreased in agranulocytes of muscles, and similar effect in agranulocytes was observed in combination of acidification and hypoxia. And so we may conclude that in general, ocean acidification combined with hypoxia or normoxia didn't promote oxidative stress, especially if we compare these results with the salinity, where the response was very quick and very substantial. And also we may conclude that mussels are pretty tolerant to water acidification, and maybe this is related to their natural habitat, since the Black Sea, as we found in the literature, really may be acidified, especially in the regions near Turkey, where the natural fluctuations of water acidity reaches is within the range of our experimental acidification. For example, when we studied this like global literature, we found that the level of 7.3 is really low for global ocean. But for the Black Sea, interestingly, is that these levels are really seasonally, naturally occur, and this is not linked, for example, to the pollution, area pollution. And monitoring research show us that it's like a long-term tendency. So maybe mussels really acclimate to this periodic acidification. So since we didn't really found any substantial change, oxidative stress at water acidification. And the last model that I will show you, it's about hypoxia. The hypoxia is my theme of research, and I started my career of as scientist by studying the effect of hypoxia on teleosts, and here I will show you the effect of short-time hypoxia on our commercially important bivalves, Mediterranean mussel and the Pacific oyster. And we did an experiment where mussels were exposed to, and oysters were exposed to hypoxia at the level 2 milligram per liter, and we sampled the parameters at the first after the 24 hours of hypoxia, and then after 3 days of hypoxia. And in some cases, for example, in mussels, we could see the effect of reoxygenation, but these results are not shown for the whole parameters. And here we've also evaluated the levels of intracellular production of reactive oxygen species, and also we evaluated the intensity of aerobic metabolism in hemocytes during hypoxia by evaluating changes in mitochondrial membrane potential in these cells. And in this experimental model, we were able to evaluate not only the activity of major antioxidant enzymes, but also we evaluated the expression of the similar genes to see how deep is the adaptive response to short-term hypoxia. So what did we see? We see that mussels and oysters differently responses to hypoxia, and the differences were seen at cellular and tissue levels. For example, in mussels, hypoxia didn't cause substantial changes in the production of reactive oxygen species during the exposure period, but we found a decrease of ROS formation after the 24 hours of reoxygenation. So this is very interesting result, and maybe it is related to POS theory, as maybe you will tell me anything about it, because really the levels of reactive oxygen species was lower at the stage of reoxygenation in both types of hemocytes. And we also found that nevertheless, despite hypoxia occurred in the environment, in water, we didn't find any changes in mitochondrial membrane potential levels. So we may conclude that the level of oxidative metabolism in mussels did not change substantially during the exposure of hypoxia. That may be indicate of their tolerance to oxygen depletion. And in oysters, the response was different. We found that after 24 hours of hypoxia in some hemocyte types and granulocytes, we observed the substantial increase of cytoplasmic ROS levels. In other cell types, it was like a tendency, but after 3 days of hypoxia, the level of ROS production decreased in all hemocyte types, and these changes correlated with the levels of with changes of mitochondrial membrane potential. Since it increased in after first 24 hours of hypoxia, and then substantially decreased after 3 days spent in hypoxia. And so we see that the paradoxical response of oysters to hypoxia is to enhancing the enhancement of aerobic metabolism in hemocytes in hypoxic conditions. And we also see that this adaptive strategy doesn't work for long periods, and even after 3 days, the level of aerobic metabolism in cells is really lower. And what about the antioxidant response in gills in this species? In mussels, we found that the activity of catalase decreased during the hypoxia, and this like a time-dependent manner of a decrease. And the activity of superoxidismutase decreased only after the first day of hypoxia. But after oxygenation stage, in both enzymes, we observed that the activity of the enzymes completely restored to control level, or even higher. But interesting picture was in the expression of catalase and superoxidismutase genes. So we found that when the activity decreased, the expression of catalase was enhanced, and the highest level of expression was observed after 3 days of hypoxia, when the activity was the lowest. And then maybe we may conclude that maybe this enhancement of expression was responsible for quick recovery of the activity of this enzyme after the first 22 hours of reoxygenation. As you see, it returned to the control level. And in superoxidismutase, we also observed that after a decrease of inactivity at first 24 hours, the further exposure to hypoxia led to really strong increase of the expression of superoxidismutase activity, and this allowed to full recovery of the activity of superoxidismutase at third day of exposure. And then, similarly to catalase, we found that at normoxic condition, after oxygenation, the expression level returned to the control levels. And in the Pacific oyster, we didn't we can't see the effect of reoxygenation, but we found that catalase catalase activity was stable during the whole period of hypoxia, but the superoxidismutase activity decreased at late stages of experiment. And these changes occurred at were accompanied with an increased, enhanced expression of superoxidismutase gene, and the expression levels were increased during the first day of hypoxia, and then tended to increase during the rest period of experiment. But this enhancement of the expression wasn't sufficient to recover the activity of superoxidismutase at the late stages of hypoxia. So, what are the general conclusions with and in comparing the response between these two species to hypoxia? We found that mussels are really tolerant to short-time hypoxia. At the cellular level, we didn't find that immunocytes didn't respond to hypoxia by changes of the cytoplasmic cross levels, and were characterized with stable levels of mitochondrial membrane potential that allows us to suppose that the level of dissolved oxygen concentration was sufficient to produce enough energy, since we also didn't find any changes in immune markers, and we found that the cellular immunity parameter were relatively stable. But we found that that hypoxia inhibits the activity of antioxidant enzymes in gills, but it rapidly restores after reoxygenation or even at prolonged hypoxia exposure. And for oysters, we see that in the position of from the hemocytes, we saw really a decrease of mitochondrial membrane potential, and this was accompanied with a decrease in the levels of reactive oxygen species production in these cells. And we also found that the activity of major antioxidant enzyme superoxide dismutase decreased, and it didn't restore even after the expression was higher, and it wasn't sufficient to restore its activity in gills during their exposure to hypoxia. And what are about our conclusions? Of course, we are here discussing the POS strategy and how it is present in various organisms. Here, by evaluating the response of two Bibalves species during the experimental modeling of environmental stress, we didn't find, for my opinion, substantial facts that allow us to hypothesize that these species show the POS strategy. But we may conclude that among these stressors, among acidification, salinity, and hypoxia, salinity and osmotic stress is the major factor that is really associated with substantial oxidative stress in these mollusks. And maybe this is linked to the strategy that bivalve mollusks use for acclimation to osmotic stress. Despite their really tolerant to salinity fluctuation and live in conditions of really different salinity conditions, but we can see that it is accompanied with substantial oxidative stress at cellular and tissue level. So I'm finishing my presentation. I hope that it was interesting for you, and I'm open for your questions.
Oh, Aleksandra, thank you for your seminar. Very nice. You present 3 sets of results. Oh, thank you. So I have a few questions. Yeah, thank you. First of all, on the work of the work on salinity, yeah,
Mm-hmm.
you use 18 parts per thousand as a control, and you study a range from 6 to 40. So in the natural environment of the Black Sea, what are the actual variations that happen in
the Black Sea? The actual variation are between I think 40 PCU till 20. The salinity is really salinity stable, but for example, in the Sea of Azov, that is close to the Black Sea, salinity is lower. It's about 10 PCU. Yes, and mussels, for example, mussels can enter like a spot during the distribution at larva station. They may enter the Sea of Azov, and it's interesting that they can live in this condition, but they are not able to undergo spawning at this, so they don't reproduce themselves in the Sea of Azov. So of course, the salinity 6 and 40 is really extremal levels, and we didn't find this levels in the Black Sea during monitoring research. But the aim of research was to find the range of natural adaptive potential of mussels.
So these variations are related to location, as you said, Azov Sea, but so it does not happen a variation during like 1 day, 2 days.
No. No, it doesn't occur. It may be like seasonal fluctuation of salinity because we have like a very hot summer and we are acidic region with very rare rains, and therefore the salinity may increase during summer a lot.
Okay. Okay. The other question is about acidification. So
Mhm.
in your acidification studies, you use pH 7.3. So it's maybe you said that, but I didn't remember. So what was the control pH?
The control pH was 8.3.
Oh, 8.3. So
Yeah,
it's quite basic. Yeah.
yeah. Black Sea is really basic, quite basic.
Oh, and again, what are the natural variations in pH?
The natural variation of Black Sea acidity really corresponds to our modeling. I don't know. As I remember, the pH level didn't increase substantially, but there are a lot of regions, especially on the south coast of the Black Sea near Turkey, where the water is really acidic. So they have regions where during summer, I don't know the reasons why it occurs, but during summer they have regions where the water acidity decreased up until 7.3, 7.2. And maybe therefore mussels really we didn't see substantial response physiological, since it's their range of tolerance.
Okay, so again, these differences in acidification, they are not temporal. They're stable over time, at least for weeks. Yeah.
Yeah, it's like seasonal variation.
Yeah. So I'm thinking about the experiments you do, but because you start with a normal a pH that they're used to, and then you decrease the pH, or you change the salinity in a short period of time. So maybe these animals, it's a condition that they're not used to, yeah, because they're in a stable condition. I mean, it's stable over weeks,
Mhm.
but changes.
I understand your question, but for salinity, we didn't change the level. The degree of salinity change was really slow. We changed it by 2 PCU per day. So and as we see during, for example, heavy storms that we had in Sevastopol this winter, or some really hot summers, the speed of salinity change, especially at low depth region, is pretty close. Maybe the period of exposure, maybe it's not. In our experiment, it lasted, for example, at about 2 weeks, and maybe it is longer period of exposure, but we didn't use a long time period of response. We want
Okay.
to find the start the processes that like initiation of the adaptive response, how the beginning stages of adaptation.
Okay. So thank you. So let others make questions. Thank you very much.
Thank you.
So it's open for everyone to make questions, please.
I have a question. So great presentation, Aleksandra. Very, very exciting news. So I have a couple of questions to ask. One of my colleagues here, we're trying to do kind of like a similar experiments, but with the mosquito larvae, basically. But it's with respect to dehydration, and then try to vitrify them. But it's kind of like similar, like, they like the salinity and stuff. Have you tested whether the amino acid composition changes during these times where like the change in salinity occurs, and whether that change in amino acid composition is what causes the pro-survival response?
No, we didn't. We didn't measure it. You mean the amino acid like adaptation to osmoregulatory response? Yes.
Yeah.
No, we didn't measure it, and it's really interesting theme for us because the process how mollusks respond to osmotic stress is still open, and we know only that at cellular level, like different experimental models, like on hemocytes, for example, we found that there are several cellular responses, but really we don't know what are the physiological mechanisms. We only studied these like processes during in vitro modeling of osmotic stress, and we found that hemocytes, similarly to vertebrate cells, they undergo regulatory volume decrease processes in hyposmotic conditions. So they swell, and then they return to their initial volume in hyposmotic water, and maybe this process allows the mollusks to function in anisosmotic conditions. But what are real osmolytes that are involved in these processes? We don't know. We may only predict that, for example, inorganic osmolytes like natrium proton exchange, potassium proton exchange or calcium chloride cotransport, like similar to vertebrate cells, they are less involved in these processes. We found that calcium, like a messenger intracellular, it is involved in osmoregulation, but we don't know which amino acids or which organic osmolytes really are responsible for osmoregulation in bivalve mollusks. Maybe one day, but I think that the major problem is that we don't have enough equipment to measure these changes.
Fair enough. Thank you so much. One other question that I have is that, so the results that you showed with the antioxidant enzymes seem pretty interesting to me. Have you done any histology to see whether the localizations of these enzymes changes from the cytoplasm into the mitochondria or other organelles?
We did histological research, and we also, for example, in the model of salinity fluctuation and in the model of acidification, we also made histological analysis which pathological alterations are in gels. We didn't localize the antioxidant enzymes, but we measured which lesions occurred during this exposure. And we found that salinity really is induced really significant changes in the structure of gels. So we found that even inflammatory responses, we found numerous hemocyte infiltration, we found the changes of lamellar structure, and even necrosis, these were many of changes, especially at extreme salinity. Salinities, they were similar in hyposalinic and hypersalinic condition, non-specific, but they are like non-reversible. But during the acidification model, we also saw like signs of inflammation in gels. We saw hemocyte infiltration. We saw some changes in the morphology, but these changes in the morphology were higher in the model where acidification was combined to hypoxia. And my opinion is that these changes are may be related to gas exchange processes, like adaptation more for hypoxia, not for acidification. But when we compare in general the pathological lesions in mussels and oysters that we saw during salinity experiment and during the acidification, salinity is like the more substantial changes occurred in gills, and the acidification was like less serious for muscles. All changes were reversible.
Awesome. Thank you so much, Aleksandra.
Thank you,
Yeah,
Hasha,
thank
for your
you.
question. Oh, Paital, please.
Yeah. Good evening to everyone this evening in India. So I just wanted to know, this was a very wonderful presentation. You did controlled laboratory experiments. So how do these animals respond in their natural environment as far as salinity is concerned?
Sorry, I didn't hear. I had a problem with connection.
Yeah. So you did a natural control experiment in laboratory. So
Mhm.
how do these animals respond in their natural environment as far as salinity is concerned?
How do they respond to natural salinity fluctuation?
Yeah,
Uh,
yeah, correct.
Really, I can't answer to this question because as I answer to Professor Marcella, the Black Sea region is really stable. Of course, our region is quite stable, and we didn't see, like, we don't have fields where the salinity periodically and we can predict the changes in, for example, in salinity fluctuation. It's like accidentally occurs, and we are now in the beginning of the stage of sampling and the monitoring of these changes to sample mussels from or oysters from this region. We have an access to capture mussels and oysters from the farm, but here I see that the conditions of salinity and acidity is really stable. As well as hypoxia. So therefore, we use the strategy of experimental modeling. And maybe in our further way of research, we will find the site where the conditions will fluctuate naturally, and where we can sample enough mussels or other model species to evaluate the natural response in our region.
Okay, thank you very much.
Okay, everyone, we still have 6 minutes because the connection is lost. So please make questions. Professor Gloria, do you have any questions? So she does a very beautiful work with several stressors in the shrimp. Okay.
Okay, if there's no other questions, so may I ask the question?
Oh, Gloria. Gloria.
Hello, good morning. Buenos dias from Mexico. Actually, I can you hear me? I'm not sure if
Yeah.
I have the microphone on.
Yes, perfect.
Okay. Actually, I had one specific question. When you talk about SOD gene expression, what SOD enzyme is this? Because there are several isozymes.
Yeah, I know. We used I can send you the primer, the primers, and they are presented in our paper that I showed in the heading. We used for superoxide dismutase, we used cellular nuclear superoxide dismutase. I think that it's like manganese superoxide dismutase, as I remember.
Okay, so it's a mitochondrial enzyme, but it's nuclear encoded because there is a Cooper zinc also, SOD. We work on shrimp, and maybe I will tell you something soon in a seminar. the results are different depending on the isozyme in expression, but it's more difficult to measure specific activity of the mitochondrial or the cytoplasms. So it's a nice work. Good to hear you guys. As I said before, it's early for us. Not too early. It's 7 in the morning when the meetings start. So actually, it's my second time that I'm joining, but I've been following in the WhatsApp and the other papers. So
Thank you.
Thank you.
Yeah, thank you for your question. Gloria, we will contact you soon to see if you can present your latest works. It will be very nice. And the same for Professor Paital. We also will get in contact with him. So is there... Okay. So do anyone has more questions? Any more questions? I have one comment on your hypoxia work. So we still have 2 minutes. And so I see that your first time point was 1 day. So maybe you missed some interesting changes in between 0 and 1 day, maybe 5 hours, I don't know, 6 hours, 12 hours. Yeah, maybe there is an increase in, for example, free radicals, in a few hours. Maybe you see more changes in antioxidants with less time. What do you think?
I think yes, maybe, of course, and I think that is hypothesis is really true, because after 24 hours we saw really substantial response, and especially if we speak about oysters, because they do not cope with hypoxia. They try to compensate it by increasing their aerobic metabolism, as we concluded from this experimental model. And yes, maybe, but I also we choose this 1 day, 24 hours hypoxia, similarly because it's really close to our day fluctuations of dissolved oxygen concentration during the summer. We found that sometimes at low depths, it really 1 day fluctuation. So maybe it's a good idea to see the response during the shorter period. Maybe we really see interesting adaptive moments.
Yeah, because some changes that occur within few hours, and then after that, the fun part is over.
Okay.
Yeah. Okay, we have only 2 minutes. Maybe we can open the so we can take a picture. So we have